do you want to?
Thank you.
Okay, we're now live. Good morning, buenos días a todos. Bienvenidos a la Masterclass de hoy con el doctor David Applin, profesor de la Universidad de Cambridge, biólogo y autor de muchos libros de texto para preparatoria, secundaria en Inglaterra. Un académico que está publicando todavía muchos papeles acerca de sus temas de biología y sobre todo el área de DNA y biología evolucionaria. Pero David nos dará más información al respecto. David, many thanks for joining us today. I've given a very brief introduction about you and your work to date. Maybe as we start the presentation, you could give a little overview of your work as a university professor and a teacher and and your publications. And we will be very ready to start in a few moments. People are joining the room, but whilst people are joining, I'm going to play a short video about St Andrew's College, Cambridge, where David Applin was the head of science for a number of years and continues to tutor at the college. So this will give you a flavour of St Andrew's College, Cambridge, where we are offering programs, pre-university programs for students looking to study at British universities. I'll play that now. Sorry, I'm just now we've seen the video, just to confirm that all the students are now joining the room. So welcome, bienvenidos a todos que se están juntando con nosotros el día de hoy. Está con nosotros el doctor David Applin de la Universidad de Cambridge, también profesor y director de ciencias en St Andrew's College, Cambridge. David nos va a dar una clase, una plática hoy acerca de lo que es su trabajo de investigación principal el día de hoy, que es DNA, que fue descubierto en Cambridge en 1953. David nos va a tocar estos temas y otros acerca de DNA y la evolución biológica. Entonces, pues por ahora, bienvenidos a todos. David me menciona que la presentación tiene bastante detalle acerca del tema de hoy, DNA. Entonces, los que estén interesados en ciencias de la vida, ciencias médicas, ciencias biológicas, que están considerando estudiar una carrera en áreas afines, sería muy útil para ustedes tomar notas durante su presentación y luego al final tendremos un espacio para hacer preguntas. Hay un chat donde pueden poner sus preguntas, sea en inglés o en español. Si en español, traduciría yo para David poder contestar. Entonces, favor de tomar notas de los puntos más importantes o más de interés para ustedes y luego pueden hacer las preguntas que quieran terminando lo que es la clase, la presentación del Dr. David Applin. So, David, I've just been explaining that students can ask questions, giving a bit of background about you, and that they should take some notes to be ready to ask questions towards the end of the class, and you'll be more than pleased to help them with those questions. So, well, without further ado, and many thanks, David, again for joining us. Over to you, David, for today's masterclass on understanding DNA.
David, many thanks for the introduction. Mexico, welcome to Cambridge on a sunny afternoon. The story today is the story of a molecule, DNA. We are living in a slow burn DNA revolution, and DNA underpins life on Earth. And so without more ado, we will be going through a slide presentation, which will consist of around about 40 or so slides. The slides will be available to you after the presentation, and you will hopefully find these slides useful preparation for any of you who are thinking of either studying the life sciences or medicine university. David has already very kindly introduced me. Basically, I first studied zoology at Leeds University. I did postgraduate work at Imperial College and then a PhD in Birkbeck College, all of London University at that time. And my particular area of research was in insect biology. I'm an author of various research papers and a number of books on biology. I was formerly head of science, as David said, at St Andrews and a member of senior management, and I now tutor for St Andrews, and I also teach in the Cambridge University, and my particular specialism, my special interest is in evolutionary developmental biology, which is the modern synthesis of evolutionary thinking. I'm an occasional poet, very amateur, I must confess, and I write and illustrate and have done stories for children. And this is in some way a distraction from biology. So let's start the presentation, and let's start with the slide that is in front of you, because the slide that is in front of you is the model represents the structure of DNA. All stories have a beginning. This story is about life's story, the story at the molecular level of a molecule, ancestral DNA or its precursor, the genetic material replicated in parent cells and inherited when they divide by their daughter cells. These cells become parent cells in their turn, and on division transfer their DNA to their daughter cells through countless divisions, giving rise to countless generations from the beginnings of life on Earth about 3.8 billion years ago, where each billion is 1,000 million years to now. I hasten to add that we use the word daughter to refer to just the new cells that arise from old cells. It has nothing to do with either maleness or femaleness. It is completely gender neutral, but it is just a word that we use to describe cells that have arisen as a result of division of parent cells. Now let us remember, proteins build the characteristics of organisms, and characteristics change through time because proteins change as DNA changes. DNA mutates through time, changes that are winnowed by natural selection from the variations in characteristics thrown up by changing DNA. Natural selection is the driver of evolution, that is, change through billions of years of time from ancestor organisms to the living things we see today. Those characteristics that proteins form, favored by natural selection, enable those organisms with the favorable characteristics to preferentially survive in changing environments. That is a very Darwinian statement about evolution. To bring it to today, understanding DNA and its close relative RNA is central to life's story, past, present, and future. Central to our understanding of life past, ancestors and their descendants alive today, including us, very much us, is DNA. Because understanding DNA and RNA as the genetic material of life helps us to grow the food we need, very much us, because understanding DNA and RNA helps us to keep healthy, understand the diseases that make us well, and indeed an understanding of death itself. The current pandemic brings the importance of understanding DNA and RNA sharply into focus. Understanding the RNA genome of coronavirus, understanding the human DNA genome as the basis of our response to the virus and the vaccines being used to protect us from the virus, some of the vaccines themselves developed and produced through our understanding of RNA technology. Our story of DNA opens in the Eagle. The pub, one of the oldest in Cambridge, stands across the street from the Church of St. Benet's and its 1,000-year-old Saxon tower. The pub is recorded in a lease dated 26th of March 1566, held by Corpus Christi College here in Cambridge, as a new build called the Eagle and Child, although parts of the building within the Georgian front date earlier to the 14th century. It was fashionable in the 18th century and the early 19th century to face buildings with a new facade facing onto the street to give them a modern look. And this is why the Eagle looks to be Georgian, that is around about 1790s to the early 1800s in architecture. But inside it is much older. The pub is popular with town gown and tourists. The Eagle was a popular meeting place for staff from the Cavendish Laboratory located nearby in Free School Lane before the laboratory moved in 1974 west to its current site off the Madingley Road. At lunchtime, 28th of February 1953, two scientists from the Cavendish burst into what was then the front bar of the Eagle and announced allegedly that we have discovered the secret of life. The we were Francis Crick and Jim Watson. Their discovery was the structure of DNA. The plaque on the wall celebrates the momentous event. Using the research data available to them, they had worked out its structure and fired the starting gun on the DNA revolution that today is changing the way we do medicine and so much more that is vital to our everyday lives. We live in a slow burn DNA revolution. Here we can see the main gate of the Cavendish through which Crick and Watson allegedly dashed on that fateful February day along Free School Lane to the Eagle, although Crick, in his book, What Mad Pursuit, has no recollection of doing so. In such ways, I guess that legends are made. On the road, someone, I guess a student, has playfully highlighted the connection between the location of the laboratory and the groundbreaking work that was carried out there. Work that continues in the new Cavendish on Madingley Road, West Cambridge. The unknown person has spray painted the formula for the base guanine, one of the components of a DNA molecule, as we just saw on a previous slide. Now for some basic structural chemistry. A molecule of DNA is made of two polynucleotide strands, each strand formed by the condensation of mononucleotides. There are four types of mononucleotide, each differing in its base, either adenine, letter capital A, thymine, letter capital T, guanine, letter capital G, or cytosine, letter capital C. The sugar rings of adjacent mononucleotides join through the phosphate group attached to carbon atom 3 of one sugar to carbon atom 5 of the next sugar in line. The links are phosphodiester bonds and hold each strand together. The strands coil around each other, forming a double helix molecule stabilized by hydrogen bonds between the base of each mononucleotide of one strand with the opposite base of the sugar of the other strand. A only bonds with T. G only bonds with C, so-called complementary base pairing. The strands are anti-parallel. One strand represented as you look at the diagram left side running from carbon atoms 3, 5, 3 of the deoxyribose pentose sugars and so on in one direction, while its partner strand right side runs 5, 3, 5 in the opposite direction. In this slide, center image, are the American biologist Jim Watson, born 1928 and still with us, and the English physicist turned biologist Francis Crick, 1916 to 2004. In 1953, with their model showing the structure of DNA, Crick is pointing to the model. Watson is looking on. Clamps hold together the spiral pattern of the model's double helix. Notice the model sugar near to Watson's left shoulder. And that model, a model of that model, is in the new Cavendish laboratory along Madingley Road. Erwin Chargaff is bottom left, whose work highlighted the importance of base pairs, although Chargaff didn't realize the importance of base pairing to the structure of DNA at the time. Bottom right is an image of Maurice Wilkins, who was working on the structure of DNA at King's College, London. Rosalind Franklin is top centre, also at King's, and Franklin provided the crucial piece of evidence that contributed to Watson and Crick's success. The scientists shown in this slide are the main players in the period around about 1951 to 53, which was the period during which the structure of DNA was put together finally by Watson and Crick. But the contributions of the other scientists were vital to Watson and Crick, and particularly the work of Rosalind Franklin. And there is a whole literature about the investigations that unraveled the structure of DNA, and that is easily available from bookshops or on Amazon. Imagine you are suspended above a spiral staircase. There's one on the end of the MRC building in Cambridge, and the MRC building stands for the Medical Research Council, research laboratories that are centred at Adam Brooks Hospital here in Cambridge. Imagine the staircase representing a molecule of DNA, and you're looking down its central axis. A bird's eye view, if you like. Keep this in mind as we continue to look at X-ray diffraction, Franklin's vital contribution to Crick and Watson's understanding of the structure of DNA and their model building. Here we see a modern X-ray diffraction apparatus, safer to use than the apparatus used by Franklin and Wilkins, and in comparison more compact, as we can see in the next slide. And here we can see the X-ray apparatus used by Wilkins back in the 1950s, and similar to the apparatus used by Franklin at King's. The caption underscores the importance of a DNA fibre being maintained at a constant humidity when the fibre is being X-rayed. Humidity affects the structure of DNA. High humidity maintains the symmetry of the B form of DNA, which was the form of DNA Franklin X-rayed for her famous photograph, which we'll see shortly. Compared with her photos of the B form of DNA, her X-ray photographs of the drier A form were harder to interpret as a 3D structure because of the asymmetry of the A form molecule in its drier state. Discovering the arrangement of atoms of molecules is possible through X-ray crystallography. The technique involves focusing a narrow beam of X-rays through a crystal or fibre of the material to be analysed. Some X-rays pass through the spaces between atoms of molecules of the material, but others collide with the atoms and are deflected. The pattern of deflections is detected and recorded as spots on film. X-rays affect photographic emulsion, or with more modern X-ray apparatus, other types of detector. The spots are called reflections. Analysing the position of the reflections and how many there are, that is their density at each position, corresponds to the position of the atoms making up the molecule. Using a type of mathematics called Fourier analysis, it is possible to work out a three-dimensional model of the molecule from the two-dimensional X-ray image of its reflections. And that is the way that Rosalind Franklin chose to go in order to try and discover the structure of DNA. She, of course, was pipped at the post by Crick and Watson. The Fourier analysis that she was undertaking is a form of Fourier analysis called the Patterson synthesis, and without modern computers, this is a very laborious process. Today, we put the information, we put the data through a computer, and the answer comes out in seconds or minutes. But it was taking Franklin weeks and weeks and weeks to data in order to try and read structure of DNA. Watson and Crick picked her at the post because they were equipped with her data and her photographs, and the photographs and the data rather of the other scientists that we saw in the previous slide. And Watson and Crick made an inspired leap and built the model of DNA which we saw in that slide. Here we have the famous plate 51 that Franklin took in 1952, and which Watson saw. He was shown this by Maurice Wilkins at King's London without, it would seem, without Franklin's permission. And therein lies a whole story of controversy, which you can research on Google, and there are a number of books about this. But remember, if you will, please, the bird's eye view diagram we've just seen. Imagine you're looking down the central axis of a DNA molecule as if a spiral staircase. Looking with a bird's eye view down the central axis means we lose the dimension of depth. The image is two-dimensional, just like the X-ray image of DNA shown here. The dark circle-like part of the image, like the handrails of a spiral staircase, positions the outer sugar phosphate groups of the nucleotides. The cross, like the steps of the staircase, locates the paired complementary bases holding the two strands of DNA together. I remember as an undergraduate at Leeds walking past a building called the Asprey Department of Biophysics on my way to the zoology department and wondering what physics had to do with biology. Little did I realize the impact of the physical sciences on biology, and this was in my youthful unknowing. Without realizing it, Asprey was, from 1938, on the way to solving the structure of DNA. That he didn't is perhaps part explained by the notion of the mind unprepared. Unlike Crick and Watson, with minds prepared, Asprey was primarily interested in the physical structure of wool, which is made up of the protein keratin. Leeds was a centre for textile research, sheep farming and wool being an important part of the local economy. DNA was something of a sideline for Asprey, also that he was refused by the university's Senate the molecular science centre he wanted, meant that the centralisation of expertise of the kind available to Watson and Crick at the Cavendish was not available to Asprey. If it had been, our story of who discovered DNA structure and where, it might be a very different story. Working with Asprey, Elwin Baton's 1951 X-ray photograph of DNA shown here reminds us of the missed opportunities of a mind unprepared. Her 1951 image of DNA predates the famous plate 51 we've just seen taken by Franklin and her PhD student Raymond Gosling in 1952. Baton's image is better resolved than Bell's image of 1938, taken in Asprey's laboratory at Leeds and which we saw on the previous slide. The central cross in Baton's X-ray, denoting the helical shape of the DNA molecule, and circle of reflections of varying density are clear, mirroring these features shown in plate 51. But Asprey did not appreciate their significance. And physicist Crick was able to interpret correctly as the helical structure of DNA. Minds prepared. This slide is a summary of what we are reviewing in the slides that follow. Genes, the genome, and chromosomes. A gene is a sequence of DNA nucleotides, each carrying one of four bases, as we have seen, A, T, G, or C. Some of the sequences encode information enabling a cell to express, that is, make, RNAs, a process called transcription, and express protein, that is, make protein, a process called translation. The genome refers to all of the DNA in the nucleus of a cell. Chromosomes consist of proteins to which DNA binds. DNA is a double-stranded helical molecule consisting of many nucleotides joined together, as we have seen earlier. Most genes in nucleated eukaryotic cells, that is, cells that have got a distinct nucleus, are not a continuous sequence of nucleotides encoding proteins. Introns are non-coding sequences that split up exons, the coding sequences of a gene. The diagram is the idea. Transcription of RNA removes the introns in a process called editing, producing mature RNA that moves from a cell's nucleus into the cytoplasm of the cell, binding to cell ribosomes in preparation for translation of protein. Introns are absent in prokaryotic, that is, non-nucleated bacterial cells. The word phenotype refers to an organism's characteristics, the result of the expression of its encoding genes, collectively its genotype. If we talk about the organism's characteristics, at the simplest, if we think of human beings, is what we look like. The color of hair, the shape of face, the color of skin. All of these are part of our phenotype. And as we've said, the term genome includes all of the cell's DNA, coding and non-coding. Genomics refers to the study of genomes. Here we see a read of a section of human DNA. The sequence of bases are represented as a sequence of small horizontal colored bars. 4 different colors, each representing one of the 4 bases. Green is equal to adenine. Blue is cytosine. Red represents thymine. And yellow is guanine. The first genome sequenced was virus 5174 here in Cambridge by Fred Sanger in 1977. He was awarded a Nobel Prize for his work. It took Sanger 2 years to sequence the virus 5174. Now, the much, much larger human genome can be sequenced in 1 day. Sequencing is automated, and bioinformatics utilizes maths and supercomputers to interpret the DNA sequence reads, making up a genome. We can see in the diagram that chromosomes are represented as bar-shaped structures, each visible as paired chromatids joined by a centromere. Under the microscope, chromosomes are visible in the nucleus of a cell when the nucleus divides. Each chromatid carrying many genes bound to proteins called histones. A complete set of chromosomes of an individual is called a karyotype. The image at the bottom of this slide shows the human karyotype. If you look closely, you can see that 22 pairs of so-called somatic chromosomes, the body chromosomes, are the same in males and females, and one pair of sex chromosomes different from one another, XX in females and XY in males. Occasionally, other combinations of X and Y can occur in individuals. In 1869, the Swiss chemist Friedrich Miescher, 1844 to 1895, discovered a substance extracted from the nuclei of white blood cells, harvested from the pus-soaked bandages he collected from the clinic near to his laboratory. He called the substance nuclein. You can see white blood cells seen in the microscope at about ×600 magnification below the photo of Miescher on the slide. Notice the large nucleus of each cell, a plentiful source of Miescher's nuclein. In 1892, writing to his uncle, Miescher speculates that nuclein could be the carrier of genetic information, but he did not distinguish between the DNA and protein histones to which DNA binds. Publication of Miescher's discovery in 1869 was delayed until 1871 by his boss, the biochemist Schuyler, who wanted to verify his, that is, Miescher's work. The article carried a note at Miescher's request that he, Miescher, had been ready to publish in 1869, protecting the priority of his discovery. Another example of the important priority, as we have seen already when Crick and Watson were first to publish the structure of DNA, basically pipping Rosalind Franklin at the post. Remember that DNA is a polymer formed by condensation reactions between mononucleotides. Its molecules carry the genetic information that enables cells to transcribe ribonucleic acid and translate proteins. Remember too, it means that the information is inherited by daughter cells when the nuclei of parent cells divide. We've already seen the slide of the graffiti sprayed on the road in Free School Lane outside the old Cavendish laboratory. Here it is again, and just let us remember that the formula is for the base guanine. I hasten to add that that graffiti lasted for about 2 weeks before Cambridge Council removed it. Perhaps unfortunate, but I guess it was a civic duty. You can see in the diagram left screen that a sequence of 3 bases, a triplet of bases, specifies which amino acid, here represented as one of the symbols shown, contributes to and its position in a molecule of sequence of 3 bases called a codon, and the diagram represents 4 codons specifying 4 of the amino acids making up part of a chain of amino acids forming a polypeptide. Let's pause here to make sure that the meanings of the words protein, polypeptide, and peptide seen on the slides are understood. When we read these words, protein, polypeptide, and peptide, tend to be used interchangeably. And one can understand why, because each of the categories of molecule are made up of amino acids. So to continue, proteins, polypeptides, and peptides, as I've just said, are categories of molecule each made up of amino acids. The difference between each category is mostly one of size of molecule and arrangement of the amino acids that make up each molecule. Molecules of protein are the largest, usually reckoned as consisting of 50 or more amino acids. Next in size at 20 to 50 amino acids, and peptides are the smallest at up to 20 amino acids. The numbers are not exact, and protein chemists may think of the numbers differently, but I give them to you here because they represent a reasonable rule of thumb estimate. The sequence of base triplets of coding DNA, codons, is the genetic code, the information enabling cells to specify which amino acids contribute to and combine in the correct sequence, making a protein. Simple arithmetic had suggested to Crick that the genetic code was a triplet code, as we have just seen, and he tells the story in his book, What Mad Pursuit? And I do recommend you to buy that. It's a fascinating insight into the life of a scientist, and also to, in particular, a perspective on the discovery of DNA. It was published in 1989 by Penguin Books, and it's called What Mad Pursuit? A personal view of scientific discovery, and I recommend that to you. In 1966, the genetic code for DNA is cracked. So ran the news headlines of the time. The three scientists, Marshall Nirenberg, Robert Holley, and Har Khorana, pictured above, carried out simple RNA molecules and added them to test tubes that contained all the reagents, that is, the chemicals and cell structures such as ribosomes needed for translation of protein. In the so-called poly-U experiment, they made synthetic RNA consisting of the sequence UUU, and the peptides made consisted only of phenylalanine. Nirenberg and colleagues concluded that the codon UUU specifies the amino acid phenylalanine. Following on from the poly-U experiment, they worked out the other codons specifying the other different amino acids in similar fashion. Take a moment to look at this diagram. Notice that base A, standing for adenine of the first codon reading left to right, only specifies the position of amino acid 1 in the protein chain and does not contribute to specifying the positions of the other amino acids 2, 3, 4, and 5. The same idea applies to the bases T, G, and C. We say that the genetic code is non-overlapping, and with the exception of some viruses, for example 5174 worked on by Sanger, the genetic code of all organisms is non-overlapping. The genetic code is also said to be degenerate because nearly all of the amino acids are encoded by more than one codon. For example, if you look at the table, the 4 bases are represented by 2 columns, left, right, and a row at the top of the table. You can read a codon, remember, consisting of 3 bases from the columns and row. You can see that the amino acid leucine, abbreviated LEU in the left side column, headed U, is encoded by 6 alternative codons. In the table, most amino acids are encoded by at least 2 alternative codons. Only the amino acids tryptophan, abbreviated TRP, in the right side column, headed G, and methionine, MET, in the U-headed column, are encoded by 1 different codon each. The codons in each diagram are set out for RNA. Notice U standing for uracil, not T. And so this perhaps would explain to you why I said 4 bases, because RNA, the near molecular relative of DNA, has got uracil as a base and not thymine. And so therefore the genetic code set out for RNA, which we can see, is a complement of the DNA codons from which. Of the 64 codons, 3 are stop codons. Stop means end of polypeptide chain. 1 codon is an initiator, which means start of polypeptide chain. Let's answer the questions on the slide. Mutation refers to a change in the arrangement or the amount of genetic material in a cell. Change may affect 1 gene located on a chromosome or part of a chromosome carrying many genes. Mutations in sex cells are inherited generation to generation. Mutations in somatic body cells are not. Gene mutations are the result of copying errors in the sequence of bases of their DNA. They DNA replication. If only 1 base is involved, then the copying error is called a point mutation. Look at the slide. Base pair deletions means that bases are lost. Base pair insertions mean that bases are added. Both types of mutation cause a frame shift. Looking in detail at the slide shows that a frame shift alters the sequence of all of the codons downstream of the mutation, altering the amino acids of the protein originally expressed by the non-mutated gene labeled normal on the left of diagram. Deletions and insertions result in the synthesis of new proteins that may not function properly or function not at all. Another type of mutation is called a substitution, where 1 base is substituted by another different base. The mutation does not cause a frame shift. In this slide, we can see a squash of root tip cells of the crocus plant at around about 600 magnification in the light microscope. The slide shows the chromosomes are stained purple with the stain acetoalcine. In the mid-19th century, the discovery of synthetic coal tar-based dyes that were more intense and brighter than the organic-based dyes used for centuries previously was groundbreaking for the study of cells. These new synthetic dyes intended for dyeing cloth were adapted for staining microscope sections, improving contrast, and therefore the visibility of microscopic structures, accelerating the discovery of cell structures. Also, better optics, including achromatic lenses, greatly improved image clarity making the optical microscope a powerful research tool. And the image at the bottom of the slide shows a microscope, a good microscope, what they look like at the end of the 19th century. In the 1880s, improved microscopes and the development of staining techniques that selectively stained particular parts of cells not only improved contrast, but also helped identification of particular cell structures. For example, acetoarsine selectively staining DNA helps to identify chromosomes. The word literally means coloured bodies, as we have seen in the image shown previously and on this slide. Following staining with acetoarsine, chromosomes appear as purple slender threads within the nucleus. Here we have another example of selective staining, this time inside the brain of a sheep blowfly. The Fuchsin stains pick out neurosecretory cells in nervous tissue. The image here is at times 1000 magnification and is a section of blowfly's brain showing neurosecretory material stained purple with fuchsin compared with adjacent brain nerve cells that do not take up the stain. In the previous slide and here, we have examples of colour guiding understanding of functional morphology. A chromosome consists of a length approximately 1.8 meters of DNA carrying many genes. The diagram represents a length of DNA binding to 4 types of protein called histones, as we have seen, labeled 2A, 2B, 3, and 4. Histones help to package DNA within the confined space of the cell's nucleus and regulate DNA activity. The basic units of packaged DNA are called nucleosomes. 2 are represented here, each consisting of an 8-histone protein core around which wraps 1.67 left-handed superhelical turns of DNA of approximately 146 base pairs. The image bottom right of the slide shows nucleosomes as core particles indicated by the black arrowheads, linked by stretches of linker DNA indicated by the white arrowheads. Linker DNA is about 80 base pairs long. The arrangement was first seen in the electron microscope in 1974. The idea of the nucleosome does not fully explain the packaging of DNA, and the diagram outlines our current understanding of the further structuring of chromosome. But I have to say that the details are uncertain. The diagram suggests that packaging DNA increases in complexity and that nucleosomes represent just the first order of packaging. The essential point to remember here is how the higher order structuring of DNA affects transcription of RNAs and translation of proteins. Processes underpinning our understanding of evolution through natural selection at the molecular level, and so much more. For the moment, remember that genes are not for characteristics. We see this written so often in books. Genes are not for anything. What genes do is they encode proteins that are the basis of the characteristics that make up an organism's phenotype. However, we often say that genes are for something because it's used often, it's described as such for just convenience. but it's something that is not to be perhaps encouraged. Historically, the idea was that one gene encoded a particular protein or polypeptide, but now we know that any particular gene can encode different proteins or part of proteins. The slide shown here is a simple summary of complex processes. Gene regulatory networks, GRNs for short, express gene products that contribute to forming the characteristics defining organisms, that is, their phenotype. The evidence suggests that the basic genetic processes are ancient. The processes built ancestral bodies in deep time, millions, even billions of years ago. Processes conserved, that is, maintained, unaltered in principle in evolutionary time, now to build the bodies of descendants. Environmental factors, for example, temperature, light intensity, affect gene expression, resulting in variations in characteristics. Here we see the result of differences in temperature affecting coloration in Siamese cats. Normally, the slightly higher core body temperature suppresses expression of the pigment melanin. The pigment is expressed in the cooler body extremities, giving the darker head, legs, and tail we're familiar with. However, if newborn kittens are raised in warmer surroundings than usual, then the dark coloration does not develop, resulting in cats that are light-colored all over. Siamese cats are, in fact, temperature-sensitive albinos. In bacteria, groups of genes, networks, encoding proteins with related functions are arranged in units called operons. The operon model of gene expression was developed in the late 50s and early 1960s by the French biologist François Jacques 2013 and Jacques Monod 1910 to 1976, shown here right and left respectively. Understanding how the operon model works helps us to understand how gene expression in general is regulated, although the presence of a membrane enveloping the cell nucleus modifies the process in eukaryotic cells. An operon is a length of bacterial DNA consisting of the operator, which binds a protein called repressor, the promoter, which binds RNA polymerase, and structural genes which transcribed mRNA. Another gene called the regulator, usually upstream of the other genes, encodes repressor protein. There are two possibilities. If the repressor protein binds to the operator, then the RNA polymerase is not able to bind to the promoter. mRNA is transcribed by the structural genes, and the polypeptide products of the operon are not synthesized. If the repressor does not bind to the operator, then RNA polymerase is able to bind to the promoter, mRNA is transcribed by the structural genes, and the polypeptide products of the operon are synthesized. Remember that transcription of RNA catalyzed by RNA polymerase is central to gene expression. The enzyme complex is similar in size to the histone core of a nucleosome. To be transcribed, DNA must pass through RNA polymerase, and the diagram gives you the idea. Since RNA polymerase catalyzing RNA transcription is similar in size, to the histone core of the nucleosome, DNA must be free of histones to allow RNA polymerase to bind to it if transcription is to take place. In the diagram, RNA polymerase is represented by a purple sphere. RNA polymerase cannot engage DNA bound to nucleosome histones. Nucleosomes are repositioned, forming a nucleosome-free region, an NFR, near to the start of the DNA to be transcribed. Notice in the diagram the purple sphere representing RNA polymerase bound to the DNA in a nucleosome-free region. Transcription of DNA can now begin. The processes described here help us to understand how genes are switched on, induced, and switched off, that is, repressed. The diagram shows the methyl groups bind to the bases of DNA, directly modifying its activity. This methylation binds DNA histone combinations tightly, i.e. condenses the combinations, with the result that proteins such as RNA polymerase and other transcription factors regulating gene expression cannot access and bind to the DNA. Overall, methylation inhibits DNA expression, but effects differ. Methylation of promoter genes inhibits their expression. The word expression here is used to describe gene activity. Methylation of suppressor genes promote their expression. The cetyl groups, AC in the previous diagram, bind to the amino acid tails of histones H3 and H4, probably altering the degree of attraction between the negatively charged phosphate groups of DNA and the positively charged histones. And they bind indirectly, modifying DNA activity. Increasing acetylation decreases the attraction binding DNA with histones, making the DNA more accessible to transcription factors regulating gene expression. Genes are induced, that is, they are switched on. Decreasing acetylation, that is, deacetylation, increases the attraction binding DNA with histones, making the DNA less accessible to transcription factors. Genes are repressed, that is, switched off. Until recently, variations in characteristics as the result of environmental factors were thought not to be inherited. The base sequence of genes encoding the characteristics are unchanged. Recent evidence suggests that environmental factors not only alter gene expression that results in variations in phenotypes, but also that the variations can be inherited, even though the base sequences of the genes encoding the characteristics remain unchanged. The term epigenetics refers to the study of phenotypic variations acquired by organisms as the result of alterations in gene expression caused by environmental factors. The reported inheritance of epigenetic variations has echoes perhaps of ideas of the inheritance of acquired characteristics. These ideas, once popular in Darwin's time, fell into disrepute with the advent of Mendelian genetics, rediscovered at the beginning of the 20th century. Perhaps molecular genetics allows a more complex understanding, modified in the light of accumulation of new evidence. The diagram shows epigenetic mechanisms and some of their consequences. When the epigenome is inherited, the environmental encounters acquired by the parental generation and represented by markers binding to DNA pass on to their offspring. Perhaps this epigenetic inheritance accounts for characteristics such as aptitude for music, ability in art and writing, and on a less positive side, a history of psychiatric disorders. Environment affects development and lifetime progress. Perhaps epigenetics is beginning to throw light on the mechanisms that drive the effects of the environment that we inhabit. Perhaps this is speculation, or perhaps the beginning of a new understanding of how life and the environment interact. Immortality lies in the DNA inherited generation from generation to generation over billions of years of life on Earth. Genes are the immortal ones, to quote a line of contemporary poetry. The quotes here on this slide by Monod and Crick underline the unity of form and function as the outcome of the inheritance of DNA from common ancestors through eons of time. In this slide and the next, we highlight milestones in the revolution kick-started in 1953 by the discovery of the structure of DNA. By the late 1960s, early 1970s, the period beginning with Misha in 1869 and gathering momentum in the 1950s of discovering the fundamentals of DNA biology was more or less complete. The molecule structure was known, how it replicated worked out, and its genetic code cracked. From the 70s, there followed exploitation and application of the classical knowledge. In this slide are example dates of important milestones in our developing expertise in DNA technology, with the major milestone of the completion of the human genome sequence at the turn of the century. And in conclusion, but progress doesn't linger at the start of a new millennium. Knowing the sequence of the human genome and the genome sequences of an increasing number of other life forms has opened the gates to a much greater understanding of how bodies function at the cellular and molecular level. With this understanding, it has been possible to develop medicines, drugs, vaccines that treat and prevent disease with increasing precision, as we are witnessing in this current COVID pandemic. Every death is one too many, and a tragedy hard to endure for loved ones. But in countries that follow the science and the global effort to provide vaccines for all, no matter how imperfect are the political actions of many, and there are many, DNA and its close relatives and our understanding of the molecules are playing a transformational role in the methods and treatments for containing the pandemic, reducing death and serious disease. And the story is only at its beginnings. It'll take time, probably decades, but understanding more and more about DNA and how to manipulate the molecule is and will transform medicine. On a larger scale still, understanding DNA is and will transform biological science. It's as if the DNA revolution is for biology what relativity and quantum theory is for physics, and indeed ultimately will be for all science. Thank you.
Well, David, thank you very much indeed. That's a tremendously detailed overview of DNA. What a history and what a continuing story you've shared with us today. Many thanks. A couple of questions appearing here that I'll forward on to you. People, I think, were interested in the link you made between life and the environment and how they interact. Could you expand a little bit on that? Where and how does that relationship take place?
Well, that is a very good question, David, and it's a very good question indeed. This link is really only at the beginning, I think, of being understood. And it all revolves around that word epigenetics, which is how the substances in the environment in which we live. And for example, when I say environment, I don't necessarily mean the big wide outside, the open air, but for example, the environment in public spaces, at home, whether or not one smokes or whether one is exposed to other air pollutants, and that the substances in these pollutants, thousands and thousands of them, of which I mentioned acetyl groups and methyl groups, that when they bind to DNA, they don't alter the base sequence of the DNA, such as in mutation, but what these substances do is they alter the activity of the genes. In other words, to use the word that I used, the expression of genes. And if the genes are either overexpressed or underexpressed, then this can have a whole series of knock-on effects on individual health, the way a person looks, and so on and so on. It's really actually only come to light, probably all of this in the last 10, 15 years, and has really only begun to really become a focus of attention maybe in the last 5 or 6. But just to illustrate that with one particular example, if we look at the cell cycle, which really describes the history of a cell from birth to death, if we look at the cell cycle, it goes through a series of stages which are controlled by genes, and these stages are called checkpoints. They either stop cells with faulty DNA progressing in the cell cycle, or they allow cells to go through. Now, if the expression of these genes is altered because of environmental factors, then of course, if the cell cycle runs out of control, what do we have? We have cancer. Cancer is basically a disease of DNA. And this is what we have learnt over the last number of decades, and which still is a puzzle, but without a doubt, we are unraveling and the problem and the effects of the environment are becoming more and more obvious. What we eat, what we drink, what we are exposed to in the air that we breathe, all of this can affect the expression of genes.
Tremendously interesting. Yeah, the field keeps growing and growing and developing. The other area where I saw there's some questions was in the area of very relevant, of course, the COVID vaccinations. You mentioned vaccinations in general. Obviously, the work on the development of vaccinations has taken place in a number of different parts of the world. And my very elementary understanding is that the vaccinations or the vaccines that have been developed work in different ways. I was unaware of the relevance of DNA and the human genome in all of this. How are those different vaccines? The vaccines are produced in different ways. Which of the vaccines do you think, or which vaccines are having most impact, and how is DNA playing a part in that?
Well, interesting. The answer to the question really falls into two categories. The vaccines in one category are being produced by traditional methods. For example, as I understand it, I believe the Russian Sputnik vaccine is being produced by traditional methods, which have been well tested, have gone through any number of trials, and are very effective. The other way in which vaccines have been produced as a result of DNA and RNA technology are the vaccines like the Pfizer and the AstraZeneca vaccines, which have been produced as a result of mRNA technology. Now, obviously, vaccines have been produced at such breakneck speed by a number of countries, but the virtue of the, shall I say, the RNA technology vaccines, their virtue is that as new variants of COVID emerge, which they will do inevitably, it doesn't matter where they are, they just happen to be called by country of origin. And I now know that WHO now calls them with Greek letters to make that less controversial. But this is just what viruses do. And the coronavirus is no different from any other virus. It will continue to mutate. Now, this is why we struggle to keep ahead of flu virus, because every year new strains of flu emerge because the virus mutates. And vaccines produced in the traditional way are produced more slowly than are the vaccines that are produced by the new technologies. So what that means is that with the vaccines with the new technologies, it means that the vaccines can be tweaked. They can be adjusted literally within weeks of the of a new variant becoming a variant of concern. And of course, those countries that can respond in this very quick fashion are those countries like the UK and America and countries in Europe that have a well-funded and have a history of investment in biomolecular science. And this is where a lot of the inequality globally is coming from, is that the countries that have got the scientific technology, and the UK has got a long tradition of investing in molecular biology. I mean, I can remember back in the 70s and 80s when the UK biomolecular sciences absolutely rocketed, absolutely took off. And it's those countries that are at an advantage in terms of dealing with variants of coronavirus. And of course, with, I would suggest, the humane obligation of making those vaccines available to the world.
Absolutely, yeah. Yes, I've been aware of, I keep in touch with the UK, even though I'm here in Mexico, and I can see that there seems to be a debate around how effective different vaccines are against the different variants. And I suppose the data coming through on those is relatively, well, as the new variants arrive, one has to wait for the data to see exactly how vaccines are performing. And of course, in the UK, we are quite advanced with the vaccination program. can the vaccines respond to the variants according to the changes?
Well, that certainly, as I understand it, is the work that's in progress at the moment. That adjusting vaccines for the possible evolution might escape the present formulations of vaccines. Pretty much aware that work is ongoing. I'm part of a COVID research group in terms of information about COVID, and I am aware that work is ongoing, and very much here at Cambridge and in Oxford.
Mm-hmm. Exactly. Well, I think those were all the questions that were coming through during the talk, David. Once again, many thanks for giving us your time today and your expertise, your knowledge, tremendous depth to the information you provided with us today. I don't know if you had any final words you wish to say before we close today's session.
Well, I wish I could say goodbye to everybody in Spanish. I fear that this would be a linguistic disaster, David. But all I can say is it's been a great pleasure to be virtually with you all. And should you, the student, should you wish to talk to your teachers, should you wish to talk to Mr. Thomas about any further information, I'm more than happy to respond. And otherwise, I wish you well with your studies in these difficult times. And it's a great, brave new world. And if you go into the biological sciences, it's the world to be, and it's absolutely fantastic. So I wish you all well, and goodbye.
Many thanks, David. Goodbye, everybody. Muchas gracias a todos. Adiós.